My name is Karin Darpel, and I'm not an ornithologist. I'm a veterinarian and a virologist, but obviously this topic is of great interest to us all. So together we will now explore the other global pandemic, highly pathogenic avian influenza virus, and why it is such a major threat to wild birds and an emerging risk to humans and mammals and other mammals. So I'm sure many of you have followed the recent news about the outbreak of highly pathogenic avian influenza, which has now reached dimensions that it poses a serious conservation impact for many bird species. And it has led to an unprecedented number of dead birds. And it's very concerning that it now, for the first time, also has reached the Antarctic. So influenza A viruses are obviously also known as major pathogen of humans. I'm actually going to quickly get the laser pointer up. Sorry for that. Most notably, the outbreak of the Spanish flu H1N1, which is believed to have led to the death of 50 to 100 million people. And it was known well before the virus itself was actually discovered. And this is where the name comes from, influenza flowing in under the atmospheric influence, because people believed beforehand already or felt that they were getting infected by something in the air. And in 1996, 1997, a new strain of avian influenza emerged in China, and from here then spread in several waves of epidemics globally. But all of these avian influenza are expressed by H and Ns. So what is this about? How do we classify avian influenza virus? Well, firstly, we classify them by subtypes. And these are determined by the two outer proteins of the virus, the hemagglutinin and the neuraminidase. And there are 16 classical types of hemagglutinin, with 2 more recently being discovered in bats, and 9 of the neuraminidase, with 2 more being discovered in bats as well. And what is also important to note is that the RNA genome is organized as 8 segments. So this is a segmented virus. But we also classify them, the avian influenza virus, by their pathogenicity. So we differentiate between low pathogenic avian influenza, which circulates in wild birds but does not cause disease, or highly pathogenic avian influenza. And in the field, these strains have so far been limited to types from H5 and H7. They cause disease in some wild bird species and in poultry. We characterize the viruses further as genetic lineages, and as an example here for H5, or as clades. And throughout the webinar, we will now also look into this. What does this all mean, and why is this actually important? So within avian influenza, certain subtypes and strain circulate specifically in their main host population. So we have the human influenza virus circulating in humans, swine influenza circulating in swine, and the avian influenza circulating in avian species. But we can see that the avian influenza virus and the avian species have a central role. Firstly, all subtypes, all classical H1 to H16 and N1 and N9 are circulating in wild birds. And from here, there are frequent spillover into poultry species. And there can also be a spillback from poultry into wild birds. But as you can also see that from the avian influenza reservoir of wild birds, there are frequent spillovers into mammal hosts as well. So why do we differentiate between avian and human influenza viruses? Well, this is a tale of different virus characteristic and host receptors. So avian influenza virus with their hemagglutinin, so their most outer protein, bind preferably to a so-called alpha 2.3 sialic acid receptor, while human influenza virus with their hemoglobin, so the hemagglutinin, their most outer viral protein, bind preferably to a 2.6 sialic acid receptor. And then these, the virus strains from avian influenza virus and human influenza virus have a different pH stability and also their polymerase, which is the enzyme that drives viral replication, has a different temperature optimum. And this is then matched in the host by the specific receptors. So within birds, the alpha 2.3 silac acid receptor for avian influenza virus dominates, while in human, in the upper respiratory tract, the alpha 2.6 silac acid receptor dominates. So for the circulation of viruses, it looks like this. In waterfowls, the 2.3 silac acid receptor is expressed in the intestinal and respiratory system, and can then be shed and circulate. In humans is a bit different. So in the upper respiratory tract, the 2.6 silac acid receptor dominates. For human influenza viruses to especially replicate in the upper respiratory tract, where it can then be efficiently shed by coughing and sneezing. But humans also express 2.3 silac acid receptors, but in the lower respiratory tract, in the lung. So if a human does get infected by avian influenza, it is so far not being efficiently shed because it's not in the upper respiratory tract, but it often leads to severe disease because it's replicating in the lungs. And then there are species like the swine, which express both receptor types, 2.3 silac acid and 2.6 silac acid, and can therefore be infected by avian influenza virus and by mammal strains, which makes them a mixing vessel for potential emergence of new viral strains. So far, all the outbreaks of avian influenza virus in humans have been caused directly by transmission from avian influenza from birds and poultry to humans. What we are afraid of, but what hasn't happened so far, is that the virus somehow adapts, that human-to-human transmission becomes possible. But that has not occurred so far. And then we also class them as low and highly pathogenic. But for whom does this actually apply? Well, the original classification comes from a pathogenesis index in chickens. So this is really defined for chickens. And again, there is an intriguing molecular process behind this low and highly pathogenic avian influenza difference. The hemagglutinin, again, is the most important protein because this needs to be cleaved to initiate the infection process. And the cleavage site where this protein can be cleaved differs in low pathogenic avian influenza strains and high pathogenic avian influenza strains. So in low pathogenic avian influenza strain, this is a monobasic cleavage site. And in highly pathogenic avian influenza strains, this is a polybasic cleavage site. And that means different proteases can cleave this HA protein. And which proteases can cleave these HA proteins and where they occur is a critical factor for the pathogenicity. So in low pathogenic avian influenza, these proteases really occur only in the respiratory and intestinal organs, and therefore only here are low pathogenic avian influenza viruses capable of initiating infection and causing disease. For highly pathogenic avian influenza, this polybasic cleavage site can be cleaved by ubiquitous proteases, which are present throughout the entire body, and therefore a systemic disease can be initiated. And low pathogenic avian influenza strains within chickens can actually also mutate to become highly pathogenic avian influenza strains. But even in birds, the clinical picture we then actually see is more complicated. So yes, highly pathogenic avian influenza strains leads to significant disease also in some wild bird species. but still it can cause infection in waterfowl species, which is often milder and slower. While low pathogenic avian influenza virus has a wild bird reservoir and mostly leads to mild clinical disease in poultry, but exception define the rule. So there's, for example, a strain of avian influenza, which although genetically it was a low pathogenic avian influenza strain, led to quite significant disease in a certain age structure of poultry. And to understand why influenza viruses are so successful, we need to understand two more tricks. So one is antigenetic drift. It is an RNA virus with a high mutation rate, and point mutations accumulate. So for example, in segment 4, which encodes for the HA protein. And from coronavirus, we now know that high mutation rate and point mutations mean that new variants emerge. And this can also occur for avian influenza. But we've already said that the genome is also segmented, and in a process of causing, when two different avian influenza strains or influenza strains infect the same cell, in a process called reassortment, they can exchange genome segments. And this means that maybe the H and N subtype might be the same, so still H5N1, but the virus is internally different. So what is happening in the current HPAI H5 global outbreak? Well, the ancestor is the H5N1 1996 strain from China. But in the following years, it became the importance of migratory wild birds became absolutely clear when it began to wave from H5NX HPAI virus strains in waves along the main migratory bird routes into Europe, also into Africa, although there is less information about the exact emergence and transmission of HPAI birds in Africa, also into North America, and now recently into South America. But it's really important to realize that this is not the same virus, and that an evolutionary process has been ongoing in the different waves, and that these different outbreak waves have been already caused by different clades of H5 viruses, and that the current global outbreak is caused by the clade 2344B. And not only are the clades different of the H5 viruses, but also the subtype. So here, for example, we can see that in the early 2020-2021, the outbreak in all birds was mainly caused by an H5N8 strain, while the current outbreak has completely shifted, or mostly shifted, into H5N1 strains. But other subtypes do exist, and this is why it's often called H5N8, because through this process of reassortment, they can exchange the segments and therefore exist as different subtypes. And the current ongoing HPAI H5NX epidemic, especially in Europe, is quite different. And one reason or one occurrence which is happening at the moment is that in the past, in a past wave, it was often a seasonal outbreak, and then it was a period of quiet. Or even in recurring outbreaks, it was a seasonal outbreak with less cases in the summer, and then it would occur again. But in the last 2 years, there hasn't really been a let-off in the summer, and it continued outbreaks of highly pathogenic avian flu were monitored and noticed in Europe even throughout the summer. And we can also see how closely the outbreak within the wild bird population are mimicked in outbreaks in domestic birds, and that there's a clear case for spillover, but also spillback from the domestic birds into the wild bird population. And in the different waves, different wild bird species were mainly affected. So the early outbreak of the last few years in 2020-21 was very much happening in waterfowl. And in the last 2 years, 2022-2023, has moved into colony breeding seabird species, mainly gulls at the moment. And again, one feature of this outbreak is that there has been an increased spillover in mammals, partly farmed minks, but also several predator species, including marine mammals. And in all areas where there have been outbreaks on mass mortality in birds, there have been corresponding outbreaks of HPAI, H5NX in mammals, driven by predatory behavior on dead or sick bird species, for example, foxes, or raccoons or bears, and also marine mammals which would feed on birds. But there have been 3 mass mortality events, one in captive minks in Spain, one in harbor seal coast in harbor seal at the coast of Maine in the USA, and one in sea lions in Peru, where the question is being raised if this was really only caused by a direct transmission from birds to predatory behavior, or if there is a component of potential direct transmission or transmission by the water and the environment. Because there was such mass mortality that there was the question if that was just driven by feeding behavior. And of course, there have also been reports of H5N1 infections in humans. And the WHO monitors human cases, for example, here from 2003 to 2023, and so far 887 cases of H5N1 human infection have been reported, including 485 deaths, which would be 50%. This obviously would be, it's incredibly severe and incredibly high. But again, this is why we went through all the introduction to avian influenza viruses. It's important to realize that just because it is H5N1 does not mean it is the same strain. And the human infection are caused by different clades of H5NX virus, because the different waves, as we've seen, were also caused by different clades. And that the infection with the current circulating clade of H5N1 viruses, 2344B, are actually relatively low. And therefore, the WHO risk assessment has classed the risk of infections with H5N1 avian influenza virus clade 2344B for the general public as low. But for specific occupations, that risk is low to moderate. Because so far, as we've heard, all outbreaks have been from poultry or bird species directly to people. So for certain occupations with high contact with birds, personal protection is clearly important. So what would need to happen for an H5N1 avian influenza virus to become a pandemic influenza virus which could circulate in humans? We have heard about point mutation, but it is important to realize that this isn't just 1 or 2 mutations, but that numerous mutations would have to accumulate to change these viral characteristic from an avian influenza adapted virus to a human adapted influenza virus. And while mutations might happen when a mammal host like a fox is infected, and that is one of the worries with these high infection in mammals, mutations might not necessarily be beneficial for transmission. So it could be that maybe a few mutations occur So the virus has evolved slightly away from being a perfect avian influenza virus, but is not yet a fully adapted mammalian virus, and therefore might actually not have an evolutionary advantage. One of the worries is always that reassortment could bystep some of these accumulations and lead to the sudden emergence of a pandemic strain. But again, just because one of the strain or one of these mutations might accumulate doesn't necessarily mean it will immediately lead to a pandemic or to an outbreak, because virus ecology is also a really important factor. So the virus will have to be able to go somewhere. Let's say it emerges in a fox. Then the question is, how much virus is being secreted? How big is the host population? Can this virus actually be passed on? What is the host contact? Would this new host actually have any contact with humans? And what would be the immunity in the host population to this emerging virus? So other factors, not just virus mutation and evolution, but also selection, are of clear importance. So what is being done to monitor HPAI outbreaks? So many countries analyze their circulating H5N1 avian influenza virus strains by full genome analysis. And here we are presenting some examples from Switzerland. So why is full genome sequencing being carried out? So this is part of molecular epidemiology, where through phylogenetic comparison of circulating avian influenza virus strains globally, and through defining the specific genotype, outbreaks can be traced, and it can be seen where they might have arisen from. And of course, full genome sequencing analysis also allows to monitor for critical mutations. And this is a collaboration between our institute and the National Reference Center for Poultry and Rabbit Diseases in Zurich, and the EU Reference Laboratory for Avian Influenza and Newcastle disease in Italy. So from selected Swiss avian influenza strains from outbreaks were sequenced, fully genome sequenced. And then, for example, phylogenetic analysis carried out for segment 4, which encodes for the HA protein, and was put into the global context to which they are most closely related, or, for example, compared within the Swiss context. And here we can see that for segment 4, this falcon is very closely related to the gull, and therefore it's interesting to speculate that the falcon might have eaten a gull. But it's also important to realize that although, for example, these two swans are quite closely related within genetically in segment 4, they still belong to a different genotype. So that means they must be different in some of the other segments. To classify the genotypes of avian influenza virus, all segments have to be compared. And for the Swiss cases, we were able to identify that 4 different genotypes are currently present. And they are very similar to the ones circulating in European neighboring countries. What can be done during HPAI outbreaks in wild bird colonies? So obviously in Switzerland, this is on a very low scale compared to the big breeding colonies along the coastal shorelines, where there has been the reports of thousands of dead birds during a single outbreak. But carcass removal seems to be the most important measure during mass mortality events in breeding colonies. And this is partly to protect the colony themselves, because certain gull species can actually pick, they're not necessarily nice, they can actually pick on sick or dead animals. And obviously you also protecting birds of prey or mammalian predator species too, which might otherwise have a feast on the carcasses. But that disposal really needs to happen with the appropriate and safe disposal methods, and also in accordance to the national regulations. And for all of us who work with birds, either through conservation projects or disease monitoring projects, it's now really important to consider that we don't become vehicles for cross-contamination through human activities and equipment between birds. So certain equipment, there needs to be thought and processes for disinfection procedures. And also personal protection equipment is important, especially when handling sick or dead birds or during activities of close contact. So then an FFP2 mask and glasses, so that there's no exposure of mucosal surfaces, is really important. And also obviously gloves, which can be well disinfected. The most outbreaks and transmission of H5N1 has happened in the poultry sector, because obviously out in the free air with wildlife colonies, there is still potentially less accumulation of the virus, but within barns and poultry facilities, the risk is potentially higher. So what is our outlook for the future, and what are the critical questions which we are all considering? Well, first of all, what will the next bird migration season bring? And then one question which we ask ourselves frequently is, how many birds actually survive an infection with H5N1, highly pathogenic avian influenza? The problem is that we don't sometimes know if, of course, not all the birds die, but it isn't necessarily clear if they survive or just didn't get infected. And if birds survive the H5N1 or HPAI infection, do they then develop and have immunity against future infections? And vaccination for wild bird does seem unlikely, but there are obviously attempts and trials and developments to consider vaccination of poultry. And then it's clearly of interest to monitor what is the importance and the effects of spillover events into mammalian species in the future. Thank you very much for listening. And now I'm really happy to take any questions, or we can have an interesting discussion.
Bonjour tout le monde. Donc je vais faire la traduction de la présentation du docteur Karin Darpel. Donc Karin Darpel est une spécialiste de l'étude des virus et de la transmission des virus de la grippe aviaire, donc l'influenza aviaire. Donc le titre de sa présentation, c'était donc la grippe aviaire, une menace majeure pour les oiseaux sauvages et un risque émergent pour les mammifères. Donc, dans un premier temps, elle a présenté un petit peu la structure de cette famille de virus. Donc les virus de la grippe aviaire ont deux protéines. Donc il y a le H qui est l'hémagglutinine et le N qui est la neuraminidine. Je suis pas spécialiste, mais c'est plus ou moins ça. Et donc en fait, avec ce H et ce N, on aura plusieurs variations, plusieurs familles de virus. Donc et il va y avoir donc des chiffres différents. Et on aura une pathogénicité différente. Donc, on va classer les virus qui sont fortement pathogènes et faiblement pathogènes. Et en fait, cette variation va se faire en fonction de la structure de ces virus. Et aussi, on va catégoriser ces virus selon qu'ils sont des virus aviaires ou des virus qui peuvent se développer chez l'humain. Et en fait, les différences, ça va être au niveau des récepteurs de ces virus. Aussi, donc la structure de ces virus va être stable à différents pH, puisque le pH à l'intérieur d'un corps d'oiseau n'est pas le même qu'à l'intérieur d'un être humain. Aussi, l'activité infectieuse va être différente en fonction de la température, puisque la température d'un oiseau est différente de la température d'un humain. Et donc, ce qui fait qu'un virus est fortement pathogène, notamment les virus influenza, c'est le fait qu'ils peuvent muter très facilement et surtout ils accumulent en fait toutes les mutations. Et donc, on a beaucoup de variants qui peuvent apparaître au fil de la chaîne de transmission. Ok. Et donc, ce qui est intéressant avec l'étude de ces virus, c'est que lorsqu'on s'intéresse, par exemple, aux épidémies de grippe aviaire, on peut voir l'évolution et l'apparition de différents variants dans le monde, et ça suit en fait les lignes migratoires des oiseaux, puisque ça va se développer dans un premier temps chez les oiseaux sauvages, puis au fil de la migration géographique, on va avoir donc des nouveaux variants qui vont apparaître et à un moment donné, on peut avoir un passage des oiseaux vers d'autres groupes d'animaux, notamment les mammifères. Et donc, ensuite, elle a présenté donc un projet assez récent, donc qui date de 2022-2023, où ils ont analysé en Suisse la séquence du virus hautement pathogène H5N1. Donc ça, c'est une analyse du génome complet en Suisse. Et donc, ils ont pu identifier 4 génotypes distincts qui étaient en circulation en Suisse. Ensuite, ils ont réalisé pendant l'été 2023 des échantillonnages non invasifs sur des colonies de mouettes et de sternes qui étaient en fait localisées sur des sortes de plateformes au-dessus de l'eau, aménagées par les hommes. Donc, ils ont récupéré des échantillons biologiques sur les oiseaux. Donc, ça pouvait être des plumes, des fesses, des sécrétions cloacales sur les oiseaux morts. Et ils ont pu voir que il y avait 44, par exemple, juvéniles et un adulte retrouvés morts et ils étaient tous infectés par le virus H5N1. Et, euh, et donc, à la fin, elle nous a donc présenté un petit peu les conseils, notamment pour éviter lorsque nous, ornithologistes, on fait des mesures sur les oiseaux ou du suivi de population, comment éviter, si une colonie est infectée, de transmettre le virus à d'autres colonies. C'est déjà dans un premier temps d'enlever les carcasses d'oiseaux morts. Donc ça va permettre aussi de protéger, par exemple, les oiseaux de proie qui vont venir se nourrir sur ces carcasses infectées, aussi protéger les prédateurs mammifères qui peuvent venir et puis s'infecter sur des souches de virus qui peuvent être transmissibles sur les mammifères. Euh, donc, pour éviter les cross-contaminations à cause justement de l'activité humaine, on conseille de porter des équipements spécialisés, à la fois pour se protéger de la transmission du virus et aussi pour ne pas infecter en fait d'autres colonies, notamment en désinfectant tous les équipements. Et enfin, pour le futur, euh, donc dans le futur, les scientifiques restent en alerte pour savoir justement en fonction des prochaines migrations si des nouvelles souches de virus H5N1 ou d'autres variants de la grippe aviaire vont arriver. Et aussi étudier combien d'oiseaux sont capables de survivre après une infection au virus H5N1, de savoir quel type d'immunité ils développent. Et dans le cas des élevages, ce qui serait intéressant, ce serait de savoir si on peut développer des vaccins. Voilà. Merci.